Hi and welcome! In this video, we're going to use a very simple Lego example to explain the concept of a haplotype. Haplotypes are often discussed, but not always well understood. Stick with us and it will all click into place. Genetics and DNA can appear complicated, so let's simplify it for the purpose of this video. Imagine DNA as Lego blocks. Each block represents a small piece of genetic information. When these blocks stick together, they form a tower. Let's call this the chromosome. For this example, imagine about 10 Lego blocks snapped together. All blocks are the same size and shape, only the colours change. Sometimes colours repeat, like 2 green blocks or 3 red blocks in a row. These repeated colours create segments of different lengths. Each coloured section is a haplotype. For example, if 2 green blocks sit next to each other, that green section is one haplotype. A haplotype is not just one gene, it's a stretch of DNA that usually stays together when passed to offspring. Every animal has 2 copies of each chromosome, one from the mother and one from the father. This means the animal has 2 versions of each haplotype. If we zoom in further, each block contains a SNP. Our cattle genotypes are a series of SNPs. In practice, we record genotypes as 0s, 1s, and 2s at each SNP position. We sum either the big A or the little a, the dominant or minor allele. Together, the series of 0, 1 and 2 values gives us the genotype of the animal. But SNPs are not inherited independently. They sit inside haplotypes, coloured blocks that mostly move together. To understand why haplotypes are so useful, we need to go back to how DNA is passed on in the first place. When sperm or egg cells are formed, the chromosomes are copied. So now you have 4 copies of each chromosome. The pairs of chromosomes line up and are briefly held together by proteins. Think of it like temporary glue points. At these glue points, segments of DNA can swap. This reshuffling of DNA is called recombination, and it must happen to create genetic variation from parents to offspring. While segments swap, large stretches often stay together. The chromosomes then separate. Each sperm or egg cell gets 1 chromosome copy, mostly intact, ready to combine with a chromosome from the other parent. Even though we measure genotypes SNP by SNP, DNA is inherited in larger pieces. Those pieces, the haplotypes, are why we can track inherited traits efficiently. Here's why haplotypes are so useful. Instead of tracking thousands of SNPs individually, we can follow these coloured blocks through a family. This block moves from great-grandparents to grandparents to parents to offspring. Even though some small pieces swap along the way, large sections usually stay together. By tracking haplotypes, we can see how traits or risks move through generations, giving a bigger picture than any single SNP could. So haplotypes let us follow inherited chunks of DNA and see which regions tend to be passed down together. That makes them a powerful way to track inheritance, and exactly why haplotypes have become so useful for tracing genetic defects. That's what we'll dive into next. Keep watching.